Good afternoon, everyone. So in the interest of trying to be as inclusive as possible, I'm not going to have any slides up on the screens. I'm going to try and speak for the next little while. However, being a basic scientist, sometimes we forget that the language that we use in the lab is not always everyday language. And so if at some point while I'm speaking I say something which is not entirely clear, please feel free to interrupt me and ask questions as we go along, because I would like this for this to be as interactive as possible. So I was invited to speak a little bit about the research that we are doing in our lab and to talk about what is sort of the status of research right now in vision. And so most of the stuff that I will focus on is gene therapy, which is the area of focus in my lab. But I'm happy to answer any questions that you may have in other areas of research in vision if there is something that you want to know. I'm not an expert in the areas of stem cells or the retinal implants, but I am familiar with what is happening right now in the field, and I'm happy to answer those questions as well. So the title of my talk is Slowing or Stopping Disease Progression by Preventing the Death of the Cells in the Retina, Our Progress Towards the Clinic. And so, for some of you who have been to Vision Quest before and have heard me speak before, I've talked about our progress towards the clinic in the past, and I know that for many patients, it becomes frustrating because it seems like we're ready to sort of introduce the therapy into the clinic, and it just takes forever to get there. And so I share your frustration. It is something that we can sort of chat about briefly at the end. It takes entirely too long to try and translate our therapies into the clinic, and I understand that. Some of the issues have to do with safety in the sense that we have to ensure that our research is as safe as possible before we can introduce it into patients. Some of it has to do with funding. Developing a therapy for the clinic is very, very expensive, and research funding is not always available. So I probably spent half my life writing grants and trying to find sort of additional sources of funding so that we can support the next phase of our research. Anyways. I'm kind of getting ahead of myself. So the one thing that we know about retinal disease is that it is a major cause of blindness worldwide, but we know very little about some of the causes of different types of retinal disease. We know that there are potentially hundreds of genes that, when mutated, can cause a disease process. But for many patients that present with a form of retinal degeneration, we still don't know which gene is the gene that is mutated in that family, or what is the specific mutation that is causing that disease. What we do know is that many forms of retinal degeneration have one thing in common, and that is that ultimately, near the sort of the final stages, when the disease starts to progress, it is because of the death of the cells in the retina, which is sort of the back of the eye. And so once those cells start to die, that is when the disease starts to progress. And the other thing that we know is that the death process is something called apoptosis. And so what do I mean by apoptosis? So apoptosis is, it's also been called programmed cell death or cell suicide. And this is a process by which an organism can remove a cell that is unwanted or that is damaged. And so apoptosis happens in our bodies all the time. And sometimes it's a perfectly normal process. And so, for example, if you've been infected by a virus, before that virus has a chance to run rampant in the body and infect all of the cells and do major damage, the cells that virus infects at first are detected by the immune system, and they are induced to die by this process called apoptosis. And by dying, by killing those specific cells that have been infected by the virus, it prevents that virus from spreading to neighboring cells. So apoptosis is very, very important for things like fighting a viral infection, very important for fighting cancer. So When a tumor first forms, the first few cells that undergo these changes that could potentially form into a tumor are often recognized by the immune system. And again, those cells that become potentially dangerous are killed by apoptosis. So those are situations where apoptosis is a good thing, but there are also going to be situations where apoptosis goes too far. And this is what we're talking about in retinal disease. So then the premise of the research that we do in the lab is that if we can slow down or stop this process of death of retinal cells, we would be able to slow down or stop the progression of the disease in the retina. And how do we do that? We use gene therapy. And so very, very briefly, the gene that we are interested in and that we work with in the lab is a gene called XIAP, which is XIAP. And what XIAP stands for is X-linked inhibitor of apoptosis. And as the name implies, this gene has the ability to block cell death, okay? And so the premise of our research is then if we use this gene and introduce it into the retina, we would be able to stop the death of those cells that would eventually go on to cause the disease. So how do we do that? So gene therapy, I'm sure many of you have heard of gene therapy. But the way that we do gene therapy is usually through the use of viruses. And so a few minutes ago, I was talking about sort of the bad things that viruses can do. Well, we can also harness viruses to do good things. And so in gene therapy, what we do is we will take a virus, we will remove all of the genes within that virus that could potentially make it dangerous, that would allow it to infect cells and kill those cells and infect neighboring cells. And so if we remove certain genes within that virus, we can actually replace those genes with good genes. And so in our case, what we're doing is we're taking our XIAP gene, which can block cell death, we're putting it inside a virus, and then we're delivering that virus into the eye. And viruses, once delivered into the eye, will do exactly what viruses are supposed to do. They will infect those cells. And so these viruses that we use for our gene therapy will infect those retinal cells and will start to produce large amounts of this XIAP protein, which can then protect those cells from dying. So today I'm going to be talking very briefly about some of our results in two diseases that we've targeted with this particular approach. And one is glaucoma, and the other one is retinitis pigmentosa. So first of all, in glaucoma, many of you are aware that glaucoma is caused by an increase of buildup of fluid in the eye. And so in the eye, fluid is constantly being produced and replaced. So that there's a little bit of sort of, there's production of fluid and then the excess fluid leaves the eye, and as much fluid as is being produced often leaves the eye, and that maintains the pressure at a normal level. But if you have too much fluid being produced and not enough leaving the eye, then you can get this buildup and you can get an increase of pressure inside the eye, and that's what causes the damage to the cells called the retinal ganglion cells that actually make up the optic nerve. And so many of you may know that then the topical drops that patients use for glaucoma are meant to prevent this increased buildup of fluid. And so some of them slow down the production of fluid, others actually increase the outflow of fluid from the eye in order to try and maintain that ocular pressure. Okay, so we wanted to test our gene therapy in glaucoma. The first thing that we had to do is we had to develop an animal model that was, that would mimic the signs of the disease in humans. And so we developed a mouse model that has glaucoma. And the way that we did that was by basically injecting something in the eye which would block the outflow of fluid from the eye. It would lead to an increase in intraocular pressure. And so we generated the model. We showed that the model behaves the way glaucoma should behave. There's an increase in the pressure inside the eye, and there is damage to the optic nerve. And so then we decided, okay, so now we've got our model, let's try and see what our XIAP gene therapy will do. So as I mentioned before, we put XIAP inside a virus, deliver that virus into the eye. It infects the retinal ganglion cells, and then we see, once we have an increased level of XIAP in those cells, can we protect those retinal ganglion cells from dying? Can we protect that optic nerve from the damage that normally is caused with glaucoma? And as you can predict by the fact that I'm standing here, yes, the answer is yes, we can. And so we were able to show then that when we delivered our Xiab gene therapy into the eye for the treatment of glaucoma, not only were we able to protect the optic nerve, so if we looked at the number of neurons in that optic nerve, we were able to preserve their number. There wasn't a reduction caused by the death of those nerves, as would normally happen in glaucoma. So we were able to preserve their structure, but we were also able to preserve their function. So not only was the optic nerve looked intact, but it also behaved the way that it should be. It functioned the way that it should be. So many of you may wonder, how can we test function in a mouse eye? Well, so the way that we can tell whether a mouse eye can see is very similar to the way that it's done in the clinic for patients. And so we use something called an electroretinogram. And so that involves placing a small little electrode on the surface of the cornea, and then you provide flashes of light or patterns of light to that animal, and you record the response, the electrical response from the retina. And if the retina is working properly, that the strength of that response will be in the normal range. If you have cell death and if you have disease progression, then you start to get a smaller and smaller response. And so by doing this electroretinogram in the mouse model of glaucoma, we were able to show that Xiab not only preserves the structure of these ganglion cells, but also preserves their function as well. Okay, and so just very, very quick summary of our glaucoma studies. So we've done this preliminary study. We've shown that Xiab protects both structure and function in the retinal ganglion cells in glaucoma. And so now the next step for us is to try and see whether we can even improve on this therapy by potentially combining it with the current standard of care. So if we use gene therapy in combination with these drops that normally lower the intraocular pressure, can we get an even better response than we normally do? And the reason why that is important is because in glaucoma patients, it's been shown that even in patients that use their drops on a regular basis, that go in for monitoring, that seem to be controlling their pressure, something is still happening to cause progression of that disease. And often those patients will still go on to lose more and more vision. And we don't really understand why that is the case, that even with controlled pressures, some of these patients will still progress. But the hope then is that if we can do a combination therapy where we're trying to control the pressure as much as possible, but also providing a survival advantage for these retinal ganglion cells, that ultimately we would be able to save those cells and prevent the progression of the disease. Okay. So that's, in a nutshell, what we've been working on in terms of glaucoma. It is one of the sort of the newer diseases that we've been working on in the lab. But the thing that I want to focus on for the rest of my talk is our studies in retinitis pigmentosa, because that is the project that is the most advanced and the closest right now to introduction into the clinic. So many of you may know retinitis pigmentosa is a disease of the photoreceptors, which are the light-sensing cells at the back of the retina. Retinitis pigmentosa is a genetic disease. It's inherited through families, and it begins with night blindness and loss of peripheral vision. Eventually, it progresses to complete loss of vision. The incidence in the general population is about 1 in 3,500. And retinitis pigmentosa can be inherited in various ways. So it may come through both parents. It's referred to the recessive form of retinitis pigmentosa. That's when both parents carry a mutation. Neither one of them is affected. But when the mutation comes from both sides and is inherited by that child, then they develop the disease. It can be in the dominant form, which means that only one parent has to be affected and carry the mutation. That parent is affected, and that parent can pass the disease on to the child. It can be in the X-linked form. And what that means is that usually there's a carrier mother. She carries the mutation, but she is not affected by the disease, but she can pass that disease on to only her sons. Okay, so there are 3 ways of inheriting retinitis pigmentosa. As I mentioned, recessive, dominant, and X-linked. And depending on which form is inherited, it's different genes that are involved. And it can also be, depending on the gene that is involved, different forms of severity. So some patients with retinitis pigmentosa start to lose their vision in early childhood. Other patients with retinitis pigmentosa can go into their 50s and 60s before they have vision loss. And so for retinitis pigmentosa, we use the same strategy. Again, ZIAP gene therapy. This time, we put, again, ZIAP inside a virus, deliver it to the retina, and this time we're targeting the photoreceptors, which are the cells at the back of the eye. We had 2 models of animal models in our lab, both of them rats. And so these rats carry mutations that cause a disease, which is very similar to the human disease. And in fact, one of the models that we were using carries the most common human mutation for the dominant form of retinitis pigmentosa. Having said that, to date, there are more than 70 genes that, when mutated, cause retinitis pigmentosa. And within those 70 genes, there are thousands of mutations. And so to try and figure out exactly what particular mutation is causing retinitis pigmentosa and to develop a therapy for that particular mutation, it would only apply to a very small percentage of patients. And so for us, using this ultimate sort of death of the photoreceptors, which is common to all forms of retinitis pigmentosa, would mean that ZIAP therapy would be applicable to all forms, regardless of which of those 70 genes are impacted and which of those thousands of mutations could be causing it. Okay, and so we treated our rat models of retinitis pigmentosa, and we found that, similar to the glaucoma case, not only were we able to preserve the structure of those photoreceptors, but we were also able to preserve their function. And so these rats, when they got the treatment, would retain their vision. And so basically, when the great thing about working in the eye in terms of research is we all have 2 eyes, and so we can treat one eye and we can leave the other eye untreated, and that way we can actually figure out exactly what our therapy is doing by comparing the 2 eyes. And so we were able to find out that in these rat models that we treated, that we saw structure and function preserved in the eye that was treated, whereas the eye that wasn't treated continued to degenerate as the disease progressed. Okay. We followed these animals for up to 7 months, and we were able to show that they still preserve their structure and their function. 7 months may not be a long time in the life of a human, but in the life of a rat, that's quite a long time, because in those 7 months, basically these animals go from being able to see almost normally to going completely blind. So we followed sort of the disease process throughout its whole lifetime, and we were able to show that our gene therapy was able to preserve those photoreceptors. And so those are the studies that we did in our lab, but we've also collaborated with other labs to see just how general is our therapy and how well will it work in other forms of retinitis pigmentosa. Because I'd mentioned to you that there are thousands of mutations that can cause retinitis pigmentosa. So maybe we got lucky, it worked in our 2 rat models, but will it work in other forms of retinitis pigmentosa? So we've collaborated with scientists, with a scientist, David Zaks, at the University of Michigan, and so he tested it in a mouse model that he had of retinitis pigmentosa that carries a very different mutation than the one that we had in our rats. And he was able to show that ZIAP gene therapy actually preserves structure and function in those mice as well. And then we've also collaborated with a scientist at the University of Pennsylvania who has dog models of, it's called progressive retinal atrophy, and that is the dog equivalent of retinitis pigmentosa. And he's tested it in 2 of his canine models, and he's found that gene therapy also preserves structure. And in one of the models actually preserved function. Now, the reason why it didn't preserve function in the other one is because the mutation causes those photoreceptors not to work in the first place. So that's sort of the drawback of our therapy. If there's a mutation that prevents those photoreceptors from functioning, from doing their job, stopping their death is not going to restore that. Okay, so only in a situation where there is vision can we preserve the remaining vision. We can't sort of cause photoreceptors that don't work to start working. And so that is the drawback of this. But for most patients with retinitis pigmentosa, they do have functional vision very early on, and then it's the disease progressions that they start to lose that vision. Okay, so we've tested now, so in our lab, we've tested it in 2 rat models of retinitis pigmentosa through our collaborators. We've tested it in a mouse model and 2 dog models of retinitis pigmentosa, and it seems to work in all of those models, suggesting that it will be a good general therapy for the treatment of retinitis pigmentosa. We've also done additional studies to look at what kind of dosage we would need in order to introduce this into the clinic, because we want to make sure that we're in the dosage range that is going to be effective but not harmful. Because sometimes if you put too much virus into the retina, you could actually cause damage as opposed to saving those photoreceptors. So we've figured out what dosage we also need in order to make sure that our therapy is working, but it's not going to cause any harmful effects. We've also done some toxicity testing with the virus, because before you can introduce any kind of a therapy into the clinic or into patients, you have to convince Health Canada and the FDA in the United States that your therapy will do no harm. And so we have done toxicity testing where you put in sort of various different concentrations of the virus. You follow these animals over a period of time. You make sure that there are no harmful effects from the injection, and that the virus doesn't escape the eye, and it sort of stays where you've delivered it. And so we've done those studies as well, and those studies are almost complete. And those will also be very important as we talk with Health Canada in order to try and get this therapy approved. Finally, we are doing something called systematic reviews, and that's basically, it's a study where you look at every single study that has been published on a clinical trial using viruses in the eye. And the reason why you want to study every study out there that has been used with every gene is because you want to identify, are there any problems with gene therapy at all? Is there a potential for a specific virus to cause a problem? Is there inflammation in the eye after you do the injection? Is there a specific dosage that works better than another dosage? What is sort of the best method for doing the injection? And by studying all of these things, then you have an idea as to what works, what doesn't, and exactly how you should structure your particular clinical trial so that it will work in patients. And so we've done all of these studies. We are now in a very good position where we can start talking to Health Canada about what is the next step. We can sort of lay out in front of them everything that we've done so far and say, is this enough? Can we now apply for a clinical trial application so that we can translate this into patients? And the other thing that we are doing right now, and as I mentioned at the beginning, I'm doing on a regular basis, is writing grants. Because gene therapy is very expensive. We've talked about Luxturna. That's been sort of the first successful treatment for a retinal degeneration. Right now, the treatment of a single patient in the US that has Leber's congenital amaurosis and is being treated with Luxturna is on the order of $900,000 US per patient. So you can imagine that the therapy is actually very, very expensive. In order to do a clinical trial, it takes a lot of money. And so we are trying to not only apply for grants in order to be able to do this, and thank goodness Fighting Blindness Canada has actually supported some of the studies that we have done. And in fact, the canine study that I talked about that we are doing with the University of Pennsylvania, we did a 6-month study with them. The FBC has now given us some funding so that we can do a more extended study, and based on that, if we can show that our ZIAP gene therapy works long-term, then hopefully we will be able to get additional funding to support the phase 1 clinical trial. And at the same time, we're also talking with specific companies that are in this area that are developing gene therapies for the eye to see whether we can partner with them in order to advance our therapy into the clinic. We are hopeful that as gene therapy becomes more common, that price tag is going to come way down. And so there are estimates that especially depending on how common a disease is, and retinitis pigmentosa is considered quite common. Glaucoma, very common disease. It can be, in certain parts of the population up to, 3.5% of the population, and it rises as people age. And so we think that the more common a disease is, the more the price tag for gene therapy is going to come down. So very, very hopeful that that over the next little while we will see a lot more gene therapies being developed for use in patients. So I want to end on an optimistic note. There are so many diseases right now. Gene therapy is starting to explode. Because of the success of the Luxturna trial, there are more and more gene therapies that are being tried for various different types of retinal disease. We think that our approach, because of the fact that it's a general therapy, would be able to treat a number of diseases, as I'd mentioned. We've tried it in glaucoma, we've tried it in retinitis pigmentosa, we have tried it in a disease called Leber's hereditary optic neuropathy, and we've looked at it in retinal detachment. It seems to save the cells in the photoreceptor in all of those diseases. And because of the fact that it's a general therapy, then we would be able to target multiple disease types by developing it. First, for retinitis pigmentosa, if it works in retinitis pigmentosa, then it would only be a very short time before we would be able to introduce it into other diseases as well. So overall, I hope to convince you that the gene therapy field is very exciting right now, and I think that over the next little while there's going to be a lot of things happening, and my belief is that Fighting Blindness Canada is going to be sort of at the center of that advancement. And that's pretty much all I had to say. I'm happy to answer any of your questions.